Hello. This exercise is an epidemic exercise. It's about the spread of a disease, and frankly, with the coronavirus going on, it seems like a good exercise. I usually do this as something to do with the reproductive system, and we treat this as an STD, which can be passed from one person to another. And the idea behind that is, or part of that is the concept that everyone you've had sex with, you're having sex with everyone they've had sex with, and so on. It originally came about, this exercise, when there were some very nasty sexually transmitted diseases that were being passed along. The exercise would have involved 12 vials of fluid, basically, or not 12, 24 vials of fluid, and basically you would have had water in 23 of those. But one of them would have had a mild acidic solution, which when you expose to phenol red, it changes from red to yellow. Now, by mixing these fluids, you're making some of them acid, and over the course of 3 rounds, interacting with three other people through the course of this, we see how the disease spreads in a group. I'm going to talk about a couple of concepts here. Basically, we're going to talk about epidemics, epidemiology, the terms, what a contagion is, what virulence is, morbidity, mortality, and so forth. So, at the end of this exercise, we're going to do an epidemiological activity where you're going to find information, find out who is sick, and basically go backwards, not so much extrapolate, but interpolate, working backwards to figure out where the disease came from. And odds are you'll only get it down to one of 2 people, because it's hard to say which person initially brought the disease into the group. Epidemiological studies are done all the time, and they are for the purpose of determining where the disease came from, what the causative factors were, so that we can figure out how to stop it. They thought at one time that malaria was caused by bad air, but in reviewing lots of data, they came to the conclusion that it was probably caused by mosquitoes. And as a result, by getting rid of mosquitoes, they were able to reduce the risk of getting malaria. They did a similar study with HIV, which came onto the scene back in the early 80s. And at that time, it was 100% fatal. And so if you got it, you were going to die. And they looked at the populations of people who actually had the disease and fairly quickly figured out, well, it's probably sexually transmitted, or a product of IV drug use. Then they went on to look at the sexual partners each person had, and they were able to do a series of interviews and say, okay, you were with this person at this time, and you were with that person at that time. Now, this person has the disease, you have the disease, the other person doesn't have the disease, and you were with the person who had the disease after you were with the person who didn't, and so forth. And they were actually able to track this down to one person, and he was an airline steward. He was bisexual, and he did international flights all over the world. They were even able to track it down to a specific time, a specific event. It was at the Tall Ships Sailing Celebration in New York Harbor in the 1976 at the Tall Ships Sailing Celebration kind of thing. And there was a big party that occurred out on Fire Island. And this guy had lots and lots of different partners, and that's where the disease hypothetically got spread. So, through epidemiological studies, we will be able to figure out what's going on here. Also, just what is an epidemic? Well, again, we are looking at a situation now where we have a virus, which in this case doubles about every 3 days, 2 to 3 days, as opposed to the one that we're going to work with, which we'll just say doubles every day. Now, in terms of a contagion, a contagion, of course, or pathogen is something that makes you sick. And it does this through virulence factors. How virulent it is how much of an exposure do you need before you're going to get sick. And there are a number of things that come into play, but for our purposes, we're going to say that also with contagion, how likely is it after being exposed that you will get sick? So this one is 100% virulent. You will get it when exposed to it. Now, there are very few diseases out there like that. The Hansen's bacillus, which causes leprosy, you have to have a load of those. I mean, you pretty much have to lick an open wound with a sore in your mouth to be able to get enough of the bacteria to actually cause you to get the disease. On the other hand, Q fever requires just 2 bacteria or 2 organisms to make you sick. So, we've got, in this case, we're going to say that this is something that does not require very much to get you sick at all. Morbidity, that's, sickness. Is it debilitating? Does it do some damage to you? Whereas mortality is how much is it likely to kill you? The mortality rate for something like the plague was in the 80s, 90%. What we're dealing with right now with coronavirus is about 3.5%. And so we're looking at, we're not going to say that this kills you. We're going to say that it injures you, it makes you sick, and we're going to look at some other activities here. Basically, what we would do is we would, again, spread the fluid, share bodily fluids. That's basically you'd take your vials and mix them together. And at the end of it, you would then, we would then test it. Now, there is a website that I want to show you, which also explains how easily something can be spread. Back in the 1800s, you had to, people didn't interact as much, a community, you might be isolated and not really meet anybody else from out of town. But with modern travel, people are all over the place, and a disease that was one place a week ago could be here today. And so I'm going to look at a concept called 6 degrees of separation. I don't know if you've heard it, but there's a game called 6 Degrees of Kevin Bacon, and I'm going to log onto a site here and show you.